and then went to Institute for Advanced Studies for a 4-year program in biology, I think, or theoretical biology in general. And he's been at NC State doing amazing stuff. And I think he's recently taken an administrative role of an interim dean of academic affairs or something like that. Yeah. So, but it's a great honor, great honor to have Alun Lloyd speak with us today on spatial spread of dengue virus, appropriate spatial scales for modern transmission. Alun.
So thank you very much, Abba. It's a real pleasure to be here. Nice to get away from, well, I guess my student issues follow me around the world wherever I am, but I am always happy to help out our students, so I'm not going to complain about that. So I should mention at the beginning, in case I run out of time, that this work, many bits of this come from a long period of time. I've been at NC State for 20 years. Fred Gould, who's an evolutionary ecologist in the area of entomology, showed up in my office in my second semester on campus and got me interested in dengue virus and the Aedes aegypti mosquito that spreads it. And so we've been working together for the last 19.5 years, and we've had a long succession of postdocs, grad students who've worked on this project. So dengue virus, so dengue is a mosquito-borne disease, and it's the most important mosquito-borne viral infection of humans. So it's a long way behind. Disease modelers like to count their importance by how many deaths or cases their infection of interest relates to. So, dengue virus is a long way behind malaria in terms of its number of deaths it causes, but it causes a large burden of disease. Dengue virus is interesting. Not unlike COVID, there's a very wide spectrum of outcomes, from a large number of people who are asymptomatic through a fairly mild infections, through to very severe life-threatening outcomes. That includes dengue hemorrhagic fever, which involves uncontrolled bleeding, like this poor guy here bleeding out of his eyes. And if you have one of these very severe forms, the death rate can be quite high. There's no real treatment except that you can basically ameliorate it. An interesting thing about dengue that I'm not going to talk about today is that there are 4 immunologically distinct serotypes of infection. So to a first approximation, you can get infected 4 times with each of those 4 different serotypes. And something that's caused a lot of interest for modelers is that there's a very complicated interaction via immunity between those serotypes. So you can get situations where having had one serotype before can give you a more severe infection the next time. And that's actually an important point, and one of the barriers that there's been to the development of vaccines against dengue, because you need to have a vaccine that works against all 4 serotypes, or else actually the vaccine can make matters worse. So, there was what do we have? We have the Sanofi Pasteur vaccine, and that's only really indicated for people who've already had a dengue infection. The effectiveness of the vaccine is different between people who've had an infection before and not, which was bad news for the vaccine manufacturers, because their target financial audience would be US tourists going to, for example, Southeast Asia. But typically we haven't experienced dengue, so the vaccine would not be indicated for those people. At least generally, there are no sort of widely used antiviral drugs. And as I said, the transmission of dengue is a mosquito-borne infection, and to a first approximation, it only goes from mosquito to people and from people back to mosquitoes. So you have to have this sort of 2-step transmission cycle. And the reason that dengue is an issue, or the reason the mosquito-borne disease is typically an issue, is that female mosquitoes need mammalian blood, or blood at least, sorry, to produce eggs. So that female mosquitoes bite, and kind of just a side effect of that is then you have the potential for transmission between humans and mosquitoes. So again, mosquito is typically infectious forever once it gets infectious until it dies. And for dengue, and indeed for Zika, yellow fever, and chikungunya, the principal culprit is the Aedes aegypti mosquito here. And the fact that you can see this big bulbous red bit at the back means that mosquito has blood fed. There is also a secondary vector, Aedes albopictus. That's a mosquito that we have quite a bit in the southeast, up the eastern seaboard in the US. But it's much less effective at transmitting dengue and many of these other viruses. Important thing to know about Aedes aegypti is that they bite during the day. So for example, you can't use bed nets. So unlike malaria, bed nets are not going to be an approach to combat dengue. These mosquitoes are highly associated with humans. They lay their eggs in human-associated water containers. So here are some photos taken out in the field in Iquitos in Peru, where we work with folks from UC Davis who have had a long-term study there. You know, people just have, I don't know, maybe flower pots and apparently toilets and things discarded in their backyard. They collect rainwater. That's where the mosquitoes lay their eggs. That's where the immature mosquitoes grow and develop. And as I said, because we don't have vaccines or antivirals, the primary way of controlling dengue is by controlling the number of mosquitoes. And so that's something I've worked on for a long time, but I'm not going to talk about the main thing that I work on in that. So I actually work on development and deployment of genetically engineered mosquitoes that have been engineered to be resistant to dengue. And that's usually what I talk about, but I won't talk about that today. So, and we'll talk a fair bit about this. There's a great deal of complexity in the biology of the mosquito, the natural history of infection, and the way that the mosquitoes and people interact. And so this then always raises the question of how complex a model do you need? So this has been one of the themes of the research in our group over the last 20 years, is the appropriate level of complexity. And of course, this is a sort of standard question in population modeling, simple or complex models. So, you know, this sort of debate goes back in population ecology to the late 60s, early 70s, where, you know, we all know the pros and cons of having simple models where we can do math, but the real world is, but they, you know, neglect lots of reality. And then you can go all the way through to having detailed individual based models, but where you typically can't do math and all you can do is pretty mindless, tedious simulation.
Don't you have in addition the problem the more it's complex will overfit your data?
Yes, yes. So that's the other problem with the more detailed models is obviously the more complicated the model, the more parameters you have. And also well, actually, I guess for all of these models, often our knowledge of biology is not as good as we would like. So one of the questions we always ask is, is a given result robust across frameworks? If not, why not? So obviously, the more robust a result is across a spectrum of frameworks, the more confidence we can have that it's a genuine result and not just an artifact of the model. On the other hand, if we see some results in one framework but not in another, that then tells us we should probe what is the issue that's causing that discrepancy, and then we can identify what the appropriate level of complexity is. Okay, so I'm going to start off with the simplest model for mosquito-borne infection. This one was originally developed for malaria. This is due to Ronald Ross, who won the Nobel Prize for elucidating the transmission cycle of malaria, and that it included mosquitoes. So this model just has two equations. So we model humans as either being susceptible or infected. We model mosquitoes as either being susceptible or infected. And there's a key point in these mosquito-borne disease models. The rate at which, so transmission depends on mosquitoes biting people. And so the rate at which mosquitoes bite humans is always assumed to be proportional to the number of mosquitoes, which makes sense. The more mosquitoes there are, the more bites that'll happen, but independent of the number of people. So the assumption is that mosquitoes need to bite because they need to get blood, and that when a mosquito tries to bite, there's always enough people around for them to find a blood meal. And so there's an asymmetry in transmission terms in these sorts of models. Transmission is proportional to the numbers of susceptible or infected mosquitoes, but proportional to the fractions of the human population that are susceptible or infected. And of course, this model, like most simple epidemiological models exhibits threshold behavior. So we can get invasion and persistence in this model if the basic reproduction number is greater than 1. And of course, that's the average number of secondary infections for a single infectious individual in an otherwise susceptible population. And so here are the differential equations. So the model further assumes we have a constant number of mosquitoes, V, for vectors, and humans, H. And in this formulation, at least in the simplest model, it ignores human immunity, but that's easy to relax that assumption. And so these are the two differential equations for the infectious humans and for the infectious mosquitoes. And just to sort of highlight a few things that are going on here. So K is the biting rate, the per mosquito rate at which they bite humans. P and Q are the transmission probabilities from mosquito to human and from human to mosquito. We have a recovery rate for humans. We assume that they recover to a susceptible state, and we have a death rate for mosquitoes. And again, just to highlight, the transmission terms are proportional to numbers of mosquitoes. So either the number of infectious mosquitoes, I, or the number of susceptible mosquitoes, V minus I, but proportional to the fraction of the human population that is either susceptible or infectious. So as I said, there's this asymmetry in the transmission terms in this model. And once we have this model, we can think about the basic reproduction number, but we have to remember we have a two-step life cycle. So we go from a person to a mosquito and then back to people. So really, I guess if Odo Diekman or Mick Roberts were here, they would insist that really what I'm talking about here is what they call the type reproduction number. So it's number of secondary infections from a person back to a person, or from a mosquito back to a mosquito, and they're the same things. So I call those type reproduction numbers, yes. So we have to multiply the number of people infected by an infected mosquito by the number of mosquitoes infected by an infected person. Thank you. That's okay. And in turn, these sort of, so we have two components, the reproduction number for the two halves of the life cycle. And for each of those components, we just do the standard thing and multiply basically the transmission rate by the amount of time that you're in the infectious state. So from a mosquito to a person, we have to multiply the rate at which that mosquito is biting, the per bite transmission probability, and the average lifespan of the mosquito. And then in the other direction, we can argue similarly by figuring out the rate at which mosquitoes bite our focal person and how long the person is infected. Multiplying these two quantities together, we get the formula down here for the basic reproduction number. Two things to note here. The reproduction number involves the square of the biting rate, because one transmission cycle involves 2 bites. and it involves the ratio of the number of mosquitoes to the number of people. And that enters because of that asymmetry in the transmission term. So this is the story from the simple model. So I guess back in the 50s and 60s, that's pretty much where the story ended.
I don't know, why does this V over H not appear squared?
Because the mosquito is just biting. So when the mosquito is infected, it's just biting people. So it's just giving rise to infections at rate K times P. But when we have an infected person, I have to figure out at what rate is a mosquito biting that infected person. So I have to take, my infected person is 1 over H. That's the probability of the mosquito bites someone. So 1 out of my H people are infected. So that's where the 1 over H comes from. K because we've got the mosquito biting rate, and V because there are V mosquitoes biting. Okay, so as we said before, there's a lot of complexity in the biology. And so there's lots of heterogeneity. So the question that has really been on many of our minds is how important are these heterogeneities? So both the mosquito and the human populations, but also the interactions between the two, are highly structured and heterogeneous. So we can think about the human population. So here we have a map from Google Maps of the town Iquitos in Peru, where we work, or where the people with whom we work. So people live in houses in this city, and they move around between those different locations. And of course, also not just between houses, between like houses and workplaces, houses and schools, that sort of thing. The mosquito population is spatially distributed across the city. One thing you notice when you go out and do surveys of mosquito numbers in people's houses, there's a high degree of heterogeneity in the numbers of mosquitoes found in different houses. So some people's houses have very few mosquitoes. Other people have a whole bunch of junk in their backyard, lots of places for the mosquitoes to lay their eggs, lots of mosquitoes as a result.
So Alun, is the assumption here that everybody has a little backyard? This is in the country.
That's right. That's a sort of place. That's a sort of location we're thinking of. I don't think I have a great picture of that. Maybe there'll be something in a minute. Maybe not. We saw a bit of a picture of those backyards earlier on with those water containers. And as I'll talk about in a minute, the interaction between mosquitoes and people, and you probably know this already, but again, we'll talk about this some more. Mosquito bites are not distributed uniformly across the human population. They're highly heterogeneous. So some people, and again, I'll talk more about this, some people attract lots of bites, more likely to become infected, more likely to transmit. So there's potential for super spreading events, and we'll characterize this a bit in a minute. So the question is, how much does this matter for the spread of dengue? So in this town of Iquitos, my collaborators have done a very detailed study of how people move around in this city. So we're interested in the spread of dengue across this city. Something I didn't tell you about Iquitos. So Iquitos is a town of about 300,000 people these days. It's in the middle of the Amazonian jungle. So basically, this is one of the largest places on Earth you can't get to by car from another significant population center. So really the only way in or out here, so it's like an ecological island, the only way in or out here is by plane or by boat up or down the Amazon. And so what my collaborators did was they put GPS units on people and watched them moving around the city. And so then they made probability distributions of the probability of describing the movements that people made. Also subdivided into different, you can't really read it here, but subdivided into different age categories. So you could see that the kids had different movement patterns from adults and from older people. Yes. So this is the thing I talked about. Mosquito bites aren't uniformly distributed across the human population. So another study that these guys did was that they looked at a number of houses and the people who lived in those houses, and they looked at mosquitoes biting the people in those houses. So how do they do this? They would capture mosquitoes that had bitten someone. So the mosquito has that sort of blood inside them. And then they do DNA fingerprinting on the blood from the mosquito. So then they can figure out which person did that mosquito feed on. And so here they are looking at body surface area. So these are all the sort of mosquitoes. These are all the, I guess these are all the people. These are all the people in the survey. Body surface area, so a measure of their size, the time spent in different houses. So they looked at a group of people and the different places they went to. So this is a 2D plot of the size of the person and the amount of time that they spent in the locations of interest. And so, you know, each of these dots here represents a person being in a given location. And then the red dots here correspond to when they found mosquito with that person's blood. So they could figure out that a given person who was in a given place got bitten by a mosquito. And so the key thing to see is that the red dots are not distributed uniformly across all of those dots. So some people tend to get bitten more often than others. And I don't know, well, I guess there was a news report on this in the last week or so. You know, mosquitoes use various cues to find people. So they, you know, they know that the things they want to bite exhale CO2. So they follow CO2 gradients, but they also follow other chemical cues. And so, I mean, I remember this as a kid. We used to go on vacation as a family, and my brother and I, you know, the children, would tend to not get bitten as much as my parents. Now it's the other way around. So mosquitoes find it easier to find bigger people because on average you exhale more CO2 than smaller people. So larger people are more likely to get bitten than smaller people. But also, as I say, chemical cues. And I always remember this, but I can never remember the way it works. I remember once reading an article that said there was a difference in attractiveness to mosquitoes depending on whether you drank beer or wine. And I can't remember which one was more attractive. So, you know, we have all these heterogeneities.
So that's, and if you eat garlic, then they don't come to you, right?
So there is some garlic story as well, which I can't remember, but yeah.
But that's also blood group, right?
There's all sorts of things. It's very complicated. But here's an example, another one of these sorts of studies where they did DNA profiling of blood got from mosquitoes in Puerto Rico, where there were about 100 residents who they identified as being connected to these houses. And they found that of all the blood they recovered from mosquitoes, 3 people accounted for 56% of those blood meals. So it's clear, you know, mosquito bites are highly concentrated on a small group of people. And so there's clear potential here for super spreading. And of course, you know, we just know that that then has important implications for control and the relative effectiveness of random control versus targeted control. So our standard tool in this area for exploring the impact of heterogeneity is the multi-type model, where we basically take the Ross model and we just subdivide, we just have different types of individuals. So we subdivide our compartments according to some one or more feature. So we could have a collection of different attractivenesses of people. We could have spatial locations. And so then we have indices on the mosquito, on the human population and the mosquito population. And now, rather than having single transmission parameters or transmission probabilities, we have a matrix of transmission probabilities. In fact, we have 2 of them because we have the mosquito to human and the human to mosquito. And so one example here would be the index might represent different spatial locations. So then these transmission parameters would would depend on the fractions of time spent in different locations. So that would be a Lagrangian formulation where we're sort of modeling movement implicitly, saying that you know, an individual spends a certain fraction of their time in one location, another location, and so on. So this is sort of very classical epidemiological framework, except we've got this two-type process. So we know exactly what to do to find things like the basic reproduction number here. We do the standard next generation approach, and we make the next generation matrix, where a matrix of the number of secondary infections of type I caused by an individual of type J. And then we have to find the dominant eigenvalue of that matrix. Now, in general, of course, that's something, you know, for a general matrix, that's not going to be possible. But if we make simplifying assumptions, we can find explicit forms for the basic reproduction number when we have certain types of mixing. For instance, when the next generation matrix is of rank 1. So one example of this, and this one goes back to Chris Dye from the 1980s, is a preferential biting model where mosquitoes allocate bites according to different fractions on different types of hosts. And it turns out you get a rank 1 next generation matrix. So you can write down the basic reproduction number, and then you can do some manipulation and put it in this form, where we get what we have is the product of the reproduction number if we had a homogeneous, well-mixed system multiplied by an inflating factor, which is 1 plus the variance of these biting fractions. So in this case, heterogeneity inflates the reproduction number compared to the homogeneous setting. So this is a standard result that we know from spread of sexually transmitted infections. It's exactly analogous to that. Abba, you had a question.
I'm going to ask, yeah, do we need a square root for the R naught to show the two generations? Or?
Well, there's a reason why I don't need the square root, right? Because I work with the type reproduction number, right?
Okay.
So for me, you know, so the difference between the R naught of the square root and not is what you consider a generation to be. So for me, a generation is a complete life cycle, whereas in the square root formulation, the generation is one transmission step.
So Alun,
I just don't like having to put square roots around stuff, so I'm happy to work with the
So question here. So is gamma a random quantity? What's the variance with respect? So how do you compute this variance here?
Well, so it could just be that you've specified a collection of numbers and you just work it. So you got like a half, a quarter, and a quarter, and you just work out the variance of those numbers. I mean, it just comes from the fact that here we've got a sum over of squared quantities, and it's just a manipulation of the formula for the variance.
Okay, so variance. So you don't mean that every one of these gammas is a random quantity. You basically.
So in this case, I've got like, you know, a certain n. I got a number n, or in this case m groups, and I've got a collection of gammas that add up to 1. So it's like the fractional. Well, maybe they don't even have to be normalized here. I forget. You just got a collection of these numbers. So, and you just work out the variance across them.
Okay, thank you.
Yeah, I mean, so the result from the previous slide is a special case of a sort of a general result from this sort of model in mathematical epidemiology. So these rank 1 matrices come from what we call separable transmission. And so the general result is that we have a vector talking about the susceptibilities of individuals of different groups and the infectiousness of different groups. So here I call that the A and the B. And in general, then in this sort of setup, the reproduction number is given by the reproduction number under the average multiplied by a factor 1 plus the correlation between the susceptibilities and the infectiousnesses multiplied by the coefficient of variation of the susceptibilities and the coefficient of variation of the infectiousnesses. And the previous result is a special case when infectivity and susceptibility are perfectly correlated. So that correlation coefficient is 1, and the square of the coefficient of variation equals variance over the mean.
So I'm missing some some spectral radius here. So don't you have, if you have this multi-types, which you have here, the spectral radius of some matrix for R0 typically?
Yes, but I'm saying I'm in a special case.
So in this case, it's a matrix diagonal, maybe?
It's a yes, I work out R0 by just summing the entries on the diagonal of the matrix. Yeah, it's sigma AI. So you've got a matrix. What is it? It's like the entries of the matrix are like AI BJ. So it's separable, and so the dominant eigenvalue is sigma AI BI. And I've maybe done something sneaky where I've divided by n because I don't care about the number of groups that there are, or something like that. So anyway, this is, and so the point is here that when we have heterogeneity, in the sort of simple case like sexually transmitted infections or the biting preference, because the heterogeneity impacts both your chance of acquiring infection and transmitting infection, the correlation coefficient is 1, and heterogeneity always increases R0. But in general, those two things need not be positively correlated. And it could be that heterogeneity actually decreases R0. But in our case, at least, the high degree of heterogeneity in biting preference means this coefficient of variation squared is much larger than 1. And so this term often dominates this term. And so the R0 expression you get if you ignored the heterogeneity can be a severe underestimate. This is exactly the same as in sexually transmitted infections. I'll just mention that you can extend this sort of analysis to think about more than 2 heterogeneities, and things get a lot more complicated when you have more than 2 coupled heterogeneities. So in fact, you can even have heterogeneity having a non-monotonic impact on R0, which is quite curious. Okay, so I'm going to now think more about human movement. So the problem of people moving around the city and transmitting dengue. And one thing you should know about Aedes aegypti is that they are rather lazy compared to, say, malaria mosquitoes. Aedes aegypti are rather lazy mosquitoes. They tend not to move very much. Particularly, they don't move much compared to people. So if an Aedes aegypti mosquito finds itself in a place where there's water for it to lay its eggs, there are people to bite, they maybe don't move much more than 100 meters over the course of their lifetime. So to a first approximation, dengue is going to be moved around a city mainly by people. So I often will simplify things by ignoring movement of mosquitoes and just focus on human movement. As I've said, there are lots of people out there who use individual-based models to look at this sort of question. It's highly computation intensive. So there are models out there for Iquitos where they will model the movement of each of those 300,000 people around the city and simulate the spread of epidemics that result. I prefer most of the time to stick with more mathematically defensible and grounded models. And we could adopt that Lagrangian model, which I mentioned earlier. And there's actually a really nice series of papers by Bichara et al. That do this. Actually, and as I'll mention in a bit, I guess, actually explicitly work out R0 expressions. But particularly when I'm thinking about stochastic models, where I'm thinking about individuals moving discretely and there actually being movement events from one place to another, I prefer a model where we actually move individuals explicitly between patches. So, okay, and also now, because I really am thinking about dengue and not malaria, I am going to use an SIR model for people. So people will recover to a recovered and immune state. And then I also have demography, so I have births of new susceptible individuals. I also have deaths from each of the classes. So this would be my model in a single patch. And now I'm going to add on terms that describe the movement of people. So for each differential equation, I'm going to add on a term that describes the departure of individuals of that compartment from our patch and the arrival of individuals of that type from all the other patches. So I've got this matrix here then of PIJs that talk about the movement of individuals between patches. And of course, I need to have a conservation rule on that. So all the individuals that leave a patch have to arrive somewhere. Oh, that's right. And also this simplest model is memoryless. So it doesn't remember the patch from which individuals came. So they move from patch 1 to patch 2. Then when they're in patch 2, they move to other patches. They don't remember that they came from patch 1. That's not a desperately good model for how people are going to move from their homes to different locations around the city. So I can make a more realistic model, which I wouldn't really care to write down, but a more realistic model that tracks people's home patches. So when they move from patch 1 to patch 2, you remember that these individuals are from patch 1. And so when they leave patch 2, we send them back to patch 1. So that's like a horrendous bookkeeping exercise because you have to, you multiply the number of states by the number of patches that you have to remember. So as I said, for this sort of model, you can do the next, you can calculate basic reproduction numbers. You can use the next generation matrix approach.
In your model where people return, do they visit random patches, but they always return to one patch?
So I have a home patch, so I don't have a workplace and a home place.
No, I mean, in the model that I've looked at, no, they will just visit other patches at random with some set of probabilities, and then they'll go back. I mean, I guess that's right. So you don't have the same individuals going to the same places. So you don't have like the repeated movement that would probably be, that would for sure actually be real, what really happens in the real world.
So it's Lagrangian, not Eulerian, right?
Well, no, it's Eulerian in that I'm actually moving people from patch to patch. But I don't remember, I mean, the Eulerian is the fact that I'm actually moving people between patches, right? So I'm not implicitly modeling the fraction of time spent in different patches.
Oh, but they do not go back to their original patch.
Well, in this one here,
it's like migration now that we model migration. They move to another patch and say they're
right. In this model, they move to another patch and then they forget that they've ever been in another. Then everything's reset. Okay. But in this model, we keep track of where they came from.
Nice.
But what we don't keep track of here, I mean, so the next step then is the fact that people have habitual movements. So not only do they return to the same patch, but they would also have a tendency to move to similar patches when they move.
Excellent.
So a person going to and from work. So of course, all that's really easy to do in an individual-based model, right? So as I said, you can do the analysis, and Bichara et al. Did this analysis. They got explicit generations for next generation matrices. They got explicit formulae for R0 in certain special cases. Basically where you have a whole bunch of symmetry so that essentially all patches are equivalent. And one thing I didn't tell you here, well, I guess the implicit question is how much does human movement impact R0? And if you do this analysis and you're in a sort of a homogeneous symmetric case, what you find is that human movement does not impact R0. And that result was rather disappointing to me, but not surprising, because of course, if you have a homogeneous symmetric situation, all the patches are equivalent. So it doesn't matter if you're individuals in patch 1 or patch 2 or patch 3, they're giving rise to secondary infections at the same rate, regardless of where they are. So it doesn't matter about them moving around.
Mobility between patches is linearized or is dependent on the disease activity. And I asked Bishara this question.
Well,
if my tendency to go to patch B depends on how many cases.
No.
So what about mosquito movement? Are the mosquitoes sort of uniform across?
So remember my big simplifying assumption here. I ignore, because it's Aedes aegypti, I ignore mosquito movement. So I'm thinking just about, because the mosquitoes are moving maybe from one house to the next, and maybe 2 houses around. That isn't going to transmit dengue across the city. So to first approximation, the movement of dengue across the city is due to people moving, not due to humans moving.
It's like a house cat. You have your house mosquito.
So Alan, sorry.
Yeah.
So if
and people and movement is nothing. I mean, disease and movement are completely unrelated. I don't even in this simplest model, maybe because most people who have dengue, it's mild or even asymptomatic. Having infection doesn't stop you from moving.
Go on to another. Okay.
Mosquito abundance per patch doesn't change.
Sorry?
Mosquito abundance. The mosquito abundance
in the simplest model, no, it doesn't change.
Okay, just for the record, I asked about the mosquito abundance per patch.
Yeah. So again, in the simplest model, I assume you have a constant number of mosquitoes per patch.
Equally everywhere.
In the simplest setting, yes. So I should say, of course, if you have heterogeneity, then human movement will impact R0, and Bishara et al.'s results show that. But in the simplest case, where everything is, everything is homogeneous, movement doesn't impact R0. But the interesting thing is, if you set up individual-based models, even under my sort of very restrictive conditions of homogeneity, simulation-based results from those individual-based models clearly show that human movement impacts the ability of the infection to spread. And if I did a Markov chain formulation of the multi-patch model, I would also get similar results. So we have conflicting results between what you see from the simulation models and that R0 analysis.
So what is the Markov chain versus differential?
Well, okay, so I take my differential equations and I reinterpret all those processes as in terms of Markov chain. So I have a rate at which individuals become infectious. I have a rate at which individuals recover. So it's just the standard correspondence between a.
Be an equivalent. I mean, you have 100,000 people. That should be the law of large numbers. Why are they different?
Okay, we'll come on to that in a minute. So that's the point. This is exactly the point. So I set up a Markov chain model that corresponds to my set of differential equations in that way. And what I consider is a human population of 20,000 people, small because of simulation, modest because the simulations are expensive. I consider different arrangements of patches, different patch configurations. So I go from having 5 patches of size 4,000. So I'm thinking of modeling the city at the level of what they call like their health districts. So I have a small number of large patches down to 4,000 patches of size 5. So I'm thinking of houses. So I'm thinking about a large number of small houses. And so that's what the different colors on this graph represent are simulation results under different patch configurations. So over here, we have a small number of large patches coming across to a large number of small patches. So this thing here is considering houses. This thing here is basically just considering areas of the city. And we have some measure of the ability of the infection to spread. So in this case, it's outbreak size in terms of the per individual probability of moving from one location to another per day.
So sorry,
yeah,
I would expect that outbreak size does depend, because if you move, you sort of effectively get more susceptible vertices at your disposal. But I would, so outbreak size and R0 don't necessarily have to go hand in hand. So.
That is true. I just happen to have shown this set of results. I also have a graph, maybe it's in these slides. I don't think it is in these slides, where I basically show transmission parameter and outbreak size. So you can see where the threshold behavior happens. And you see similarly, the threshold happens for different values of the transmission parameter. So and so remember the sort of differential, the R0 sort of analysis would tell us that the movement parameter doesn't actually matter. But here we see the movement parameter does matter. And so what's going on? The R0 type analyses, it's always initial behavior, right? So you assume a fully susceptible population. So it's basically linearizing transmission. So essentially, you always set the number of susceptibles equal to the patch size. And the point is, for dengue, when we're thinking about transmission on the scale of the city, there's this appropriate spatial scale. The house is the appropriate spatial scale on which mixing happens. So the house is the natural scale at which to model things. And we simply always have small numbers of individuals on that scale. So a house has 2, 3, 5, 10 people. A house doesn't have 1,000, 10,000 people. So the point is, what goes on in this setup is that basically transmission gets to infection gets to a house, you get spread within the house, and then you get spread to the next house. So you have that intrinsic spatial scale of the house that is sort of irreducible, not in the mathematical sense, but there's that irreducible biological component of transmission. And so this sort of dynamics are never going to be captured by that sort of deterministic R0 type analysis. Even though we can set patch sizes to be small, I mean, those deterministic models simply don't respect the fact that individuals are discrete. And so, in a house of size 5, loss of one susceptible has a pretty large impact on transmission in that house. So for me, who has spent a large chunk of my career since my PhD, basically analyzing these deterministic multi-patch models, this was really unfortunate because it means that my standard go-to analytic tool is much less useful than we would hope when we're understanding transmission of dengue at the within city scale. So of course, this is an important thing to know because there are all these papers out there in the literature that claim you can use these models for that, but I am not convinced of their utility. So this then put me in a rather, not desperate, in a rather pessimistic situation that these people who do these large-scale individual-based models really were the winners in this story, so to speak. And I wasn't entirely happy to concede that fact. So I think, the plea at the time was to find.
I wouldn't concede because I would think that if you just can't analyze this thing by looking at sort of time zero. So you have to run your differential equation long enough that you see the depletion within the patches. And then this should actually start to.
I'm not sure. So, well, for example, if you look at final outbreak size, I don't think you'd get that. I don't think you would get the distinction because the point is in the deterministic model, a fraction, an atto-human can move, an atto-infected human can move from one patch to the next, seed infection there. So I am not, and then it would burn through that patch, but then again, you get very small transmission to somewhere else and it would burn through. I actually had thought that I really do need to put in the corresponding deterministic results. And I think we would be disappointed. We would be similarly disappointed if we actually looked at final outbreak size of the deterministic model.
You don't think you could model that by having different time scales? That's sort of your. Because if your infection would sort of run out in your patch faster than your movements, then you should.
I don't think that happens in a deterministic model because, as I say, small fractions of individuals can go from one patch to another, and then once it gets there, your local R0 is greater than 1, so it's going to spread. It'll take longer, but it's going to spread.
So in a sense, you're in the random environment. You have this stochastic effect at the level of your.
Exactly. That's exactly the point. We can't appeal. It's exactly because the house is the appropriate unit in which to model things. We can't appeal to a law of large numbers here.
Right. So that would be very intuitive, Alun, don't you think? I mean, once you figured out that that's the sort of level of
yes. Okay, but then of course the question is, what do we do? And so then, so there is a tool that people use to look at spread of infection where you have a household, a population that's structured in terms of households. And there are these so-called households models that go back to the 60s and 70s that have been really successful in the directly transmitted infection world. So have been used heavily in modeling the spread of flu and common colds and so on. But these models had never before existed in the vector-borne disease literature. And so some collaborators of mine in Australia, Josh Ross, Andrew Black, came up or extended these households models to mosquito-borne diseases. And so these models are formulated at the level of households and looks at the spread of households. And as part of that, you have to have a model of the within household spread of infection. And in this world of households models, the users concede that the R0 formulated at the individual scale is no longer the appropriate quantity to look at in terms of a transmission threshold, but they have something called R star, which is R0 basically evaluated the household size at the household scale. So you use the household as the unit on which you're looking at transmission. So we have a paper that's currently under revision that's up on the archive, and I'll just give a brief flavor of it. Basically, it's a branching process model. It makes a whole bunch of assumptions, at least in the very first formulation of it, but most of these can be relaxed. We assume, again, that people move, mosquitoes don't. Introduction into households is relatively rare, so we only have to think about single introductions rather than multiple introductions. We consider a collection of households at the moment of households of the same size, each of which has N people and M mosquitoes. We can relax those assumptions. And so then in this framework, you have two sorts of infected household characterized by how infection was introduced. And so either a susceptible person from your household has gone somewhere else and been bitten by an infectious mosquito, or an infected person from elsewhere has visited your house. So as I say, we have two sorts of infected household, and then use a two-type branching process that describes transmission and looks at the number of houses of each type that are caused by a house of a given type. And then this approach implicitly involves looking at the final size distributions within the two sorts of household.
So should I think of the households as, so N is large, but the number of people in a household is always small?
In this case, we have a large number of households, all of which have the same number of people and the same number of mosquitoes, yeah. In this very simple version of the model.
The same number of people doesn't go to this.
That's right. We always have a finite and small population size at the household level, both a finite number of small number of people and a small number of mosquitoes. Yeah.
And should I think of the household? I have a household and a bipartite graph with all the mosquitoes. Or is
no. So remember, for us, mosquitoes don't move. So basically, we have mosquitoes staying in a household. So we just have a collection of households, and we have movement of people between the different households.
But you do not really model that a person gets bitten by a mosquito. Do you model that stochasticity or yes?
Yeah, at the level of a household, yes.
So that's what I said. In the household, I can imagine.
Oh, in the household, yes.
Having a complete bipartite graph between people.
Correct. Sorry. I thought you were talking about the between households. Yes. No, completely.
Could
yes.
you, for simplify, assume I had just one mosquito for my whole household?
Could do. You could do.
But your model has sort of four people in the household, 10 box mosquitoes, there are 10 mosquitoes for our house. Something like that. Yeah. As I say, these assumptions can be relaxed. So we could have a distribution of human household sizes and a distribution of mosquito numbers for different houses as well. But we haven't done that yet, but that's a fairly simple generalization. I mean, at least that generalization has been made in the directly transmitted household, the model, and it's not all that much extra complication. Okay, so that's me finished. So as I say, simple models can give us important insights, but it's really important for us to understand the limitations. So heterogeneity can be really important, and we can at least imagine that we can use those simple models to give insight into the importance of heterogeneities. But we run up against this issue of spatial scale of transmission that limits the utility of the sort of standard go-to analytic tool. As I said, individual-based models have their devotees, but these models aren't the most satisfying to use. And I think this households model is a pretty promising new analytic tool, and we're hoping it's going to be able to provide new insights into, for example, the role of human movement on the spread of dengue. And so just finishing up with the acknowledgements. As I said, a whole bunch of people at NC State. The mosquito folks, generally at UC Davis and at Emory, we were part of a large NIH project grant that was looking at impact of disease and at impact of disease on movement and on transmission. And then the folks at Adelaide and various people who funded this over the years. And that's me done.
Excellent. Okay. Thank you, Alun, for a fantastic talk. Any questions? Yes, sorry.
For a very interesting talk. One thing that in mosquito, particularly dengue, factor plays is environmental variability. The generation time of the mosquitoes are tightly related to temperature precipitation gradients.
Right.
I don't know the layout of Iquitos, but yeah, assume that there are mountains or mountain ranges nearby or something like that. So these create different altitude gradients, right? That even though your patch model could be very simple and homogeneous in terms of the structure of the model, the conditions for transmission in the different patches at different altitudes or different regimes of precipitation temperature may alter that homogeneity and give you a correct result, even for a simple framework that you have. So have you considered this climate variability?
Well, okay, so I mean, what I didn't show you was the full horror. So we have, even though I spent most of my time saying they're dreadful things, we have basically an individual-based mosquito model that actually looks at, it's just horrendous. I even just get shivers thinking about it. That actually, we have a resource, temperature, weather-driven mosquito model. So we actually look, because in the main project, which is on the spread of these genes through mosquito population, we really care about the population dynamics and genetics of the mosquito across space. So yes, we have a model that looks at that. Actually, in Iquitos, it's fairly homogeneous across the city. And actually, because it's pretty equatorial, there's not so much variation across the year as, for example, when we try to model this in Australia, where it's highly seasonal. And of course, the other thing which you well know is that it's not just the mosquito that's impacted by temperature and so on. It's then the virus within the mosquito. So that's a really important thing, that the amount of time it takes the virus to incubate within the mosquito is highly sensitive to temperature, which is one of the concerns about global warming, that these vector-borne diseases, not only will the mosquitoes find themselves elsewhere on the planet, but the transmission cycle will shorten, because it takes 5 days or 4 days rather than 7 days for the virus to incubate inside the mosquito.
Thank you.
Thank you for that nice talk. I'm interested, I was interested in your discussion of heterogeneity, about how you decoupled the susceptibility and the infectiousness.
Yeah.
I'm used to thinking of those things as being correlated, basically to do with numbers of contacts.
Right.
So that your CVA and your CVB would be basically the same thing.
Right. Yes.
What in this disease, or generally in other infections, what is it that causes those to become separate? Is it within host effects, or is it effects of the contact patterns or the movement?
Right. So as you say, if the heterogeneity is a sort of a contact heterogeneity, typically the susceptibility and infectiousness will be proportional to each other. That correlation is 1. But it could be, I'm trying to think of examples. It could be like biting heterogeneity would be the same as that, because mosquitoes, it's more like transmission, it impacts transmission in both directions. But people also think about other things. There could be just differences in infectiousness across the human population. And that then wouldn't be correlated with the propensity of the mosquito to bite the people, for instance.
Within host viral load.
Within host viral load, something like that. Yeah. I mean, duration of infection is something else that people have thought about as being heterogeneous across a population. So there's all sorts of factors. So, depending on which factors you're thinking about, there would not necessarily be any need for them to be positively correlated. We actually have a paper sort of setting out that. I mean, obviously that slide or two was very sort of theoretical and obviously not directly related to the rest of them in terms of the grounding in biology. We have a sort of a theoretical paper that lays out this sort of multiple heterogeneity framework. And we spent a whole day sort of talking about different heterogeneities and whether they might be positively or negatively correlated or not correlated at all. And I think there are some comments on that in that paper. So it entirely depends what facets we're thinking of in terms of transmission.
Well, thank you very much for your talk. So I have a question. So at first you were mentioning the fact that there were 4 serotypes, and then it doesn't appear anymore. So
yeah. So
can we have this kind of feature also included in the model, or?
Yeah, you can. Obviously, if you have 4 serotypes, you have to, at the very least, multiply, each of our infectious compartments by, you used to have 4 times as many infectious compartments to track the, the different infections. Then you also have to track in humans.
But you cannot be infected twice by the same.
But yes. You can be infected twice by different things. So what you also have to track, unfortunately, is people's infection history. So if you track everything, so I've had, and maybe even the order in which you've had your infections could be important. So, if you do the full 4 serotype model, your number of states or number of equations increases dramatically. There are some simplifications. So one of them is that your body only remembers your previous infection. So, but then of course you wouldn't get permanent immunity to a given serotype. So, basically, it's, again, really easy to do in an individual-based model, makes the differential equation models really complicated. Again, it's only, complicated it's just bookkeeping. So it's not sort of conceptually difficult, it's just practically difficult. And so, I just take the coward's way out and I think about, imagine we've got a population where there's only 1 serotype of dengue circulating. So I can think about just 1 serotype.
Can we do also a comment? So it seems that you are many times opposing deterministic dynamical systems and individual-based models. But individual-based models are not only like simulations. You can have, as Etienne, for instance, showed in the first day, or as Greg has done. So you can have stochastic processes, really mathematically describing individual-based model, and that allows you to also to link between branching processes, approximations, dynamical systems. So I would not see everything so opposed, but
right. So this is my career, right? I go back a long way, and started off doing deterministic things, and then I do stochastic things. So I have a lot of papers on the correspondence between the deterministic and stochastic models. And it's a tool that I use in lots of settings, but I'm just saying in this particular case, where the household is the most important scale on which transmission happens, we are in a rather difficult place. I mean, then of course I move to looking at things on the scale of households, and then I use a branching process model. So I mean, it's all good in the end. But I'm just saying that we need to, you know, be mindful that we can't just take standard deterministic tool, and and it might be completely obvious in retrospect that we can't do this, but this doesn't stop the fact that there have been tens of papers, at least in the literature, doing precisely that in very recent years. So you know, it's one of these things. Sorry.
Agent-based and stochastic is really not the same.
No, completely, yeah.
So, I mean, when people model every single person in the city, I would call that agent.
Right.
But when you do your stochastic households,
yes,
that is
Monte Carlo or stochastic. This is just a Monte Carlo Monte Carlo simulation of a Markov
chain
model.
Actually often do give us insight. So I mean,
well, I mean, I get clear insight here in the fact that the scale on which I model things has a major impact. Yes.
But sometimes I understand that you cannot do like averaging inside the household because it's very small.
Yeah.
But if you have a lot of households, so even if you have a stochastic behavior, like in small household, they can average, if your unit is the household, not the individuals, then
well, that's exactly, yeah. So I mean, that's exactly the point, right? Yeah. The households model is formulated at the unit in the households model is the household. But the sort of standard deterministic thing just has patches, and you know, it has no notion of the fact that there's something special about a patch of size 5 or 10. That's the point. So
you shouldn't think of the household as a patch.
Well, that's what I'm saying. That's what people do.
No, but in the deterministic model, I would almost think of the household as so. If the household has 4 people, I think of it as a unit which has 2 to the 4 states, depending who is infected, or maybe 3 to the 4 states. And then you can describe a differential equation which describes how this more complicated individual, which now has more internal states, interacts. And maybe it's not a differential equation that may
right, sure. So
I mean,
let's
not worry about that.
Right. I mean, of course, ultimately, I can just write down the Kolmogorov equation for the whole thing, right? But that's a ginormous system.
No, I don't want you to do that.
Well, I'm glad you don't want me to do that.
No, I want to think. So I want you to think of the household, if it really only has 4 people, as something which has sort of 2 to the 4 states or so. And there's sort of some, in reality, it's not just exponential times and stuff, but maybe you can approximate it inside, sort of, you describe it now as transitions inside this, but it's its own state. So I don't want you to think of the household as a continuum in your differential equation, but then you have sort of, now you place each household becomes a new individual in your differential equation model, except that each individual now has more states to take into account the richness of the household. No, you seem not convinced.
I mean, I think this is not unlike what we're doing in the households model, to be honest.
But that would again give you a differential equation if you like them so much, or maybe integral equation.
Here we're really, I mean, in the households model we have a branching process because we're thinking about invasion, right? So is it going to go from this household to the next household and so on? But yes, there could well be a differential equation, I guess, there for households.
You were not here a couple of days ago, but we basically talked about these sort of Markovian models with frailty, or precisely with the type of heterogeneity of susceptibility and infectiousness. And we discussed briefly how these can be decoupled sort of in a canonical way. And it kind of seems like that might be a strategy. I think that's what Tran is saying, and what Christian is saying is kind of maybe the part of the discussion that we had. So, you know, if you were to formulate the Markov model at the level of a household,
yeah,
and you somehow could incorporate in a sensible way these heterogeneity that you...
I think that's exactly what happens in household models. That's what I'm trying to say. I think that's exactly what happens.
we are saying the same thing.
What I'm trying to say is I don't think there's anything new here, because I think that's precisely what was done back in the 70s in these households models.
But you can basically just look at the Markov process, right? Which is a household level Markov process. And for that process, you will have some kind of law of large numbers that would take into account the frame.
Across household, right. Yeah, but that's, yeah. So
rather than doing the branching process between households, you do the law of large numbers between households. But obviously the 5 is not a law of large numbers. So you have to keep the 5 discrete, and you will pay for that by having a higher dimensional state space.
Yes.
But you still should be able to do a law of large numbers,
well, of course,
and not have to do a branching process for the spread between households.
Well, except if I'm thinking about initial spread, right? Where there is stochasticity.
You want to understand the stochastic, sure.
Yeah. I mean, well, whenever we think about invasion of infection into a population, you know, invasion starts off with one individual, you know, one household. So then we, you know.
Yeah, so, you know, there's this colleague of mine, Eben Kena, who basically looks at household studies and who believes that the entire epidemiology is about households. So he would love your talk. And I think that—
Well, it's a shame Tom isn't here, because, you know, Tom's work connects to that as well, you know, so yeah.
So much fun. I like this, but I'll ask a question. So for dengue, okay, the Aedes mosquitoes prefer to bite during the day. So, and I would assume that people tend to spend more time outdoors than indoors, right? What's the average time I spend at home, for example, in the morning? It's not a lot. So the transmission is really taking place outdoors, isn't it? So I'm playing devil's advocate, you know. If you say it's all about households.
I mean, so of course, Ketos.
Oh, it's different.
Yeah, it's different. It's different. So I think most. I mean, the feeling there is that most of the transmission is happening indoors.
So people stay at home most of the time during the day.
Right. People are inside. Yeah.
I see.
Okay. Well, and the mosquitoes tend to be inside because that's where the people are. So, you know, these mosquitoes tend to bite like, you know, maybe also earlier in the day and later in the afternoon. So I think it's when people are around. I mean, maybe that's why they bite at that time.
And the problem with the dengue mosquito is it doesn't, unlike malaria mosquito, it doesn't need a lot of water to lay eggs, right? So it's very easy to get breeding habitats. Yeah, that's very good. Okay, any other question? I think we should let Alun go. This is fantastic presentation and very nice discussion afterwards. Thank you so much, Alun, for a very nice presentation. Thank you. Okay. All right, it's lunchtime now, I believe. Yes, right.